My name is Melis Olcum, and I am a postdoctoral research fellow in Center for Cardiovascular Genetics in University of Texas Health Sciences Center at Houston. I would like to present you our latest paper published in Journal of Cardiovascular Aging. Our paper is titled as Panoptosis is a prominent feature of desmoplakin cardiomyopathy. As the title implies, in this study we focused on molecular pathogenesis underlying desmoplakin cardiomyopathy, which is a subtype of arrhythmogenic cardiomyopathy. Arrhythmogenic cardiomyopathy is a hereditary form of cardiomyopathies characterized by cardiac arrhythmias, cardiac dysfunction, and premature death. Mutations in genes encoding intercalated disc proteins, especially desmosome proteins, play a major role in pathogenesis of arrhythmogenic cardiomyopathy. Desmoplakin is one of them, which causes a distinct phenotype with cell death, myocardial fibrosis, and inflammation, and therefore the phenotype is referred to as desmoplakin cardiomyopathy. Our main objective was to understand the pathogenesis of desmoplakin cardiomyopathy. We used cardiac myocyte-specific tamoxifen-inducible MerCreMer mice to conditionally delete the desmoplakin gene in the post-natal cardiac myocytes in mice. Tamoxifen was injected for 5 days starting at 2 weeks of age, and the phenotype was analyzed at 4 weeks of age. Conditional deletion of both copies of the desmoplakin gene resulted in almost complete loss of desmoplakin mRNA and protein. Deletion of desmoplakin led to extensive remodeling of intercalated discs, as evidenced by the changes in expression levels and localization of other desmosome proteins, PKP2, DSG2, DSC2, and JUP. Gene set enrichment analysis based on the RNA sequencing data from cardiac myocytes show that desmoplakin deletion led to activation of the TNF-alpha and FKB pathway, inflammation, receptor-mediated cell death programs, and epithelial mesenchymal transition, while suppressing oxidative phosphorylation and myogenesis. As the gene set enrichment analysis predicted, receptor-mediated and mitochondria-mediated apoptotic pathways were activated. Caspase-8 and its downstream target caspase-3 were upregulated, both in protein and mRNA levels, along with other genes involved in the receptor-mediated apoptosis such as TRAF4 and 6 and death receptor 5. In contrast, TROI was downregulated significantly. BCL2, an anti-apoptotic marker, was reduced, and BAD, a pro-apoptotic protein, was upregulated. Other genes involved in mitochondria-mediated apoptosis such as BAX, BID, and NOXA were all upregulated significantly. Collectively, our findings indicate activation of that receptor and mitochondria-mediated apoptosis in the desmoplakin-deficient mice. Necroptotic markers, RIPK1, RIPK3, and MLKL, and pyroptotic markers, gasdermin D and ASC, were upregulated, indicating activation of additional cell death programs, which are collectively referred to as panoptosis. ZBP1, Z-DNA RNA binding protein 1, acts as a sensor protein, which is considered as the master regulator of panoptosis. In our model, both protein and mRNA levels for ZBP1 were upregulated significantly for both isoforms, as we see with our exon-specific expression analysis. Upon deletion of desmoplakin, hearts showed extensive fibrotic areas which was spread throughout the ventricles. Heart weight to body weight ratios for these animals were markedly increased, as well as more than a dozen of fibrosis-related genes were upregulated in these mice, further verified by the increase in protein levels of TGF-beta. Median survival age for desmoplakin-deficient animals was around 2 months, while wild-type and MerCreMer control animals exhibited a normal survival. At the age of 4 weeks, mice showed severe cardiac dilatation, as echocardiographic indices show. Expression of heart failure markers showed a consistent profile as cardiac hypertrophy and dysfunction. Desmoplakin deficiency led to increase in prevalence of ventricular arrhythmias, including premature ventricular contractions and ventricular tachycardia. There were no sex-dependent differences in cardiac function and survival. Our study indicates postnatal homozygous deletion of the desmoplakin gene caused severe early-onset DCM characterized by fulminant panoptosis, severe fibrosis, and increased expression of the pro-inflammatory genes, which are the phenotypic features of desmoplakin cardiomyopathy. The precise mechanisms by which desmoplakin deficiency leads to panoptosis is unclear. Likewise, the mechanisms responsible for the increased expression of ZBP1 and whether upregulation of the ZBP1 protein is solely or partly responsible for panoptosis in the present model is unknown. The findings set the stage for subsequent mechanistic studies to delineate the molecular pathogenesis of desmoplakin cardiomyopathy. The model also provides the opportunity to test the effects of pharmacological and genetic interventions in preventing or attenuating cell death, fibrosis, and inflammation in desmoplakin cardiomyopathy.